Herkese merhabalar. Bugün Nano Open Webinarı'nda sunum Yarı Zamanlı Araştırmacılarından Koç Üniversitesi Tıp Fakültesi Öğretim Üyesi Profesör Doktor Devrim Gözüaçık konuğumuz olacak. Geçtiğimiz günlerde Moleküler Hücre Biyolojisi ve Hastalık Biyolojisi alanındaki çalışmalarıyla TÜBİTAK Bilim Ödülü almaya hak kazanan Profesör Doktor Gözüaçık, Kanserde Yenilikçi Terapötik Yaklaşımlar için Nano-Biyoteknolojiler hakkında konuşacak. Gözü açık, Horizon-MSCA-COFUND kapsamında SUNUM koordinatörlüğündeki doktora sonrası araştırmacılara yönelik bir hareketlilik burs programı olan Nano-Biotechnologies projesinin de araştırmacıları arasında yer alıyor. Devrim Hocam hoş geldiniz. Sözü ve ekranı size devretmeden önce hakkınızda kısa bilgi aktarmak istiyorum katılımcılarımıza. Doktor Gözüaçık, Hacettepe Tıp Fakültesinden tıp doktoru derecesiyle mezun oldu. Biyokimya yüksek lisans derecesini École Polytechnique'te ve Paris Sud Üniversitesi'nde, kanser hücre biyolojisi konusundaki doktorasını Paris Pasteur Enstitüsü ve Necker Çocuk Hastanesi Araştırma Merkezi'nde tamamladı. Ardından Weizmann Bilim Enstitüsü'nde kanser ve hücre ölümü konusunda doktora sonrası araştırmalar yaptı. Sabancı Üniversitesi'nde 14 yıllık akademik kariyer sonrasında Koç Üniversitesi Tıp Fakültesi ve KUTAM'a araştırma profesörü olarak atandı. Doktor Gözüaçık aynı zamanda Uluslararası Hücre Ölümü Derneği'nde yönetim kurulu üyesi ve New Mexico Üniversitesi AIM Biyomedikal Mükemmelliyet Merkezi'nde afiliye üye olarak görev yapmaktadır. Doktor Gözüaçık'ın araştırmaları kanser kök hücrelerinde, kanser stroma ilişkisinde, tümör bağışıklığında, kanser metastaz ve uyku hali yanında nörodejenerasyonda otofaji yolakları ve otofaji anormallikleri konularına yoğunlaşmaktadır. Hocam tekrar hoş geldiniz. Ekran paylaşımını size devrediyorum.
Hoş bulduk. Çok teşekkür ediyorum organizasyon için, davet için. Ekranı paylaşıyorum. Her şey yolundaysa devam edeyim. Görüyor mu?
Görüyoruz hocam.
Tamam. Because there may be foreign students here with us, so I'd like to continue in English. I hope you can hear me.
Duyuyoruz.
Okay. So, thank you again for the invitation and nice introduction. I currently work in Koç University, but we are located in the Koç University Hospital in Topkapı. So this is our hospital building. And as you can see here, there's a middle tower. In fact, this middle tower is currently almost completely dedicated to biomedical research in our university. So my lab is also located here. But of course, there's the campus in Rumeli Feneri in the northwest of Istanbul. We are affiliated, or we are part of this campus as well. So I have several affiliations and duties, but I'm also a senior part-time researcher in SUNUM Nanotechnology Research and Application Center. In addition to my faculty position in Koç University Medical Faculty and KUTAM Research Center. So our field is autophagy. So in all eukaryotic cells, there's a biological phenomenon called autophagy, which is active. From yeast to man, you see this basic mechanism. So this is one of the major degradation mechanisms in cells. When we say degradation, in general, we understand protein degradation, but autophagy is also important for degradation of lipids, sugars, and several other molecules. Because in contrast with the general protein degradation system, which is the ubiquitin proteasome system, which is responsible for the degradation of short-lived proteins and in general soluble proteins, autophagy is able to deliver its cargo to the lysosome. So in the ubiquitin proteasome system, as the name implies, the proteasome is the degradative system. In autophagy, it's the lysosomes. So in lysosomes, you can degrade all kinds of molecules, including, of course, proteins, okay? But more than that, you can also degrade whole organelles like mitochondria. So this is one of the biggest advantages of autophagy. Autophagy is important for degradation of not only organelles, but also large protein aggregates and long-lived proteins, okay? So naturally, I mean, consequently, abnormalities or malfunction of autophagy result in different diseases or aggravate some diseases. So, for instance, myopathies, muscle diseases, or neurodegenerative diseases, liver, heart diseases, infectious diseases, because it serves as an inborn cellular defense mechanism against parasites and bacteria. But on top of that, in the last, I would say, 10 years maybe, there's an increasing amount of research dedicated to understand the role of autophagy in cancer. So, of course, all these applications and importance of autophagy led to Yoshinori Ohsumi, our colleague and friend, to win the Nobel Prize of Physiology and Medicine in 2016. So it's quite a basic and important mechanism. So we focus on the connection with cancer, and there are several, of course, phases and steps of cancer. In the very early phases where cancer is forming, so normal cells become cancerous cells. Autophagy seems to serve as an anti-cancer mechanism, protecting from cancer. Because it's for example, it's the major degradation mechanism of mitochondria, which are in fact a source of reactive oxygen species. So elimination of this is alleviating the burden of genotoxic stress, so mutational burden in cells. So it's an anti-cancer mechanism. But once a cancer cell is established, then the role is changing. So autophagy is also a stress response mechanism. It's activated by different types of stresses, including amino acid, serum starvation, glucose starvation stress, which you see, in fact, in especially fast-growing or metastasizing cancer cells. And also in response to chemotherapeutic agents or different types of chemicals, you see activation of autophagy. So therefore, autophagy is involved in invasion, metastasis, spread of cancer, as well as in therapy resistance to chemotherapy, radiotherapy of tumors. And in recent years, in the last couple of years, a role in tumor dormancy is arising. So tumor dormancy, I'll talk a little about it at the end of my talk. We are very much interested in it and very excited about the results that we are obtaining. So tumor dormancy is in fact a mechanism where in fact tumor cells stay dormant or not replicating quiescent state, and they come back after a time. So this is one of the major reasons why cancer is difficult to cure. So this is called, in the medical terms, relapse or recurrence, meaning cancer coming back. Okay. So in our lab, we mainly focus on three different aspects of cancer. First is diagnosis, because we do a lot of screens and we obtain several molecules which might serve as a diagnostic marker. And I'll come to the connection to nanotechnology and nanobiotechnology. So we especially in collaboration with engineers and chemists and bioinformaticians, we try to understand what markers there are for cancer, what novel markers we can discover, and how can we use them for early diagnosis. Because as you see here, all these people are saved, these blue people, out of 9 out of 10, maybe more. when cancer is diagnosed early, but then late diagnosis result in only one person maybe to be saved when all the rest are doomed to die. So early diagnosis is not a myth, it's a reality. In fact, it's important for saving lives. So we focus on that aspect as well. So for this purpose, we try to find early diagnosis markers of cancer. On the other hand, as I mentioned, autophagy is involved in drug resistance or other treatment resistance in cancer. So we are also focusing on this aspect of autophagy in our lab. But recently, most of our efforts are focusing on metastasis, the role of autophagy in metastasis and relapse, because as I said, this is one of the major reasons why cancer is killing, in addition to drug resistance. Okay, so we focus on metastasis, spread of cancer, and relapse, and the role of autophagy. So we have interesting results about all these topics. So maybe in the coming slides, I'll mention some of these results. So I'm getting quite senior these days. Also with the career awards and everything, I feel as a quite senior person in research in Turkey, because we established our laboratory in 2006 after 11 years of study and scientific career abroad. I came to Turkey and we didn't have much at that time in the beginning. So we started with simple yeast-2 hybrid screens, cheap and simple. And already this was one of the first screens using autophagy genes, trying to catch, new autophagy-related proteins and signaling pathways and everything. And we published several public papers using these results. And in collaboration with our colleagues, we discovered one of the first microRNA groups regulating autophagy, some of the very first microRNAs. That's why we are kind of known in the field about microRNA and autophagy, and we still continue this line of research in our laboratory. And we perform several other functional screens. And lately, we have also access in collaboration with Jörn Dengel from Switzerland. We are performing mass spectrometry analysis of different interactions, and we also use some other techniques. So all this gave us tons of names of genes and proteins which might play a role in autophagy regulation and which might connect autophagy to disease. Okay, so some of them are listed here, these publications. We might want to maybe check them or read them. So this is a brief summary of some of our discoveries and contributions to the field of autophagy. So we discovered this is the autophagy pathway. So briefly, in fact, is mTORC1 is a master regulator of amino acid availability in cells and protein synthesis. When it's on, protein synthesis is activated. It's anabolic, but then when it's off, and it's very sensitive to different types of stress that also activate autophagy, including starvation, drugs, cytokines, and different things. So inhibition of this pathway leads to activation of the autophagy complexes, such as ULK1 complex, Becklin VPS34 complex. And then all these result in formation, de novo formation of membranes that you see like tongue here. And this involves ATG5, 12, 16 proteins, ATG4 protein as a cleavage protein for LC3. And the LC3 or LC3-like proteins, they are conjugated to lipids and they elongate these membranes and play a role in the growth and closure of autophagic membranes. They also have a role in the fusion with they play also regulatory roles in fusion with the lysosomes. Autophagy was long considered to be a non-selective phenomenon. So these vesicles, they form and they engulf things. These are special vesicles with double bilayered membranes, and they engulf things in the cytosol. as I said, including aggregated proteins, mutant proteins, organelles such as mitochondria or other organelles, and long-lived proteins and other things. So it's a cytoplasmic cleaning mechanism, if you will. But then the vesicles close, and then they fuse with the lysosomes, and the contents are degraded inside the lysosomes. And these capturing of targets can be non-specific. They can capture whatever comes on their way, these membranes, growing membranes, or they can capture in a selective manner. And this is called selective autophagy. And this has been studied recently intensively, and there are several receptors which link the LC3 proteins on the membranes, this MAP-LC3, which is lipidated and bound to the membranes. Here they are also present, of course. So they bridge between autophagosomes or autophagic vesicles and cargo. So we can count P62, NBR1, NDP52, and other receptors in this category, okay? So we made several discoveries in this sense. We discovered, I'll mention some of the discoveries in my talk. For example, we discovered several cytokines and their effects, their role in cell survival, cell death, cancer formation, activation, and so on. We discovered several microRNAs controlling the Becklin complex, ATG4, and other proteins in the TORC pathway. And we discovered feed-forwards signaling mechanisms. We discovered different protein complexes regulating mitochondrial autophagy. In addition, we discovered DNA response and autophagy connections, direct connections, some connections to diseases, including cancer. So we did a lot of work in the last, I would say, 25 years or so. Okay. So microRNAs, in fact, they are short RNAs. They are regulatory RNAs. And this is the central dogma. You see the DNA here, and then the messenger RNAs and proteins. So microRNAs, they regulate the availability of mRNAs. It can lead to their degradation, sequestration, or blocking their translation. But the end result is that proteins, which are in general functional units, they are regulated. So microRNAs this way regulate protein availability. So presence, amount of proteins. In this way, they change signaling pathways, and they have dramatic effects in cells. Okay, and they act as they are like around 3,000 or so microRNAs in men. So they can have several targets, but in our experience, we see that in a couple of key targets are important in each pathway. Okay, so they regulate these pathways, and they change the signal, like a cogwheel, which is in the Swiss watch. So if you take a cogwheel, or in this case a protein, then this whole signaling system is changing. Okay, so so just to give you a few examples about our research in microRNAs. So we discovered that a transcription factor called MITF or MITF was activating autophagy. But then this activation, we discovered that it passes through a microRNA. It's called miR211. And on the other hand, miR211 itself is causing nuclear translocation of these factors, for the transcription factor MITF. You see here the nuclei are empty, but then here the transcription factor is cytosolic, and then it goes to nucleus when you put the microRNA. To cut the long story short, we discovered that, in fact, the microRNA is directly targeting a component of the mTOR complex. There are two complexes. One is upstream, one is downstream. So this is called mTOR2. The Rictor protein is a component of this. So we discovered that this is the regulation of Rictor and switching off of this complex, or downregulation of this complex, result in decreased AKT phosphorylation. And and decrease AKT phosphorylation and mTORC activation. This serves as a switch off signal for the mTORC1, as does taurine 1 or starvation. Taurine is a chemical which inhibits the kinase in this complex. And then inhibition of this complex allows this transcription factors, which are in fact initially phosphorylated by the complex, to be dephosphorylated because the complex is inactive. They migrate then to the nuclei and activate gene expression. And among the genes that are expressed, there are genes which are involved in lysosome proliferation, but also genes which are involved in autophagy, such as ATG10 and LC3B. We show that there is a direct binding of the factor to the promoters of these genes. And in addition to the promoter of the microRNA. So it creates a feed-forward signal where, in fact, when autophagy is activated, then by inhibition of the TORC pathway, it stimulates its activation through microRNA. Okay, so this might be a point where, in fact, you can impinge upon for treatment approaches as well. Another hint about the potential is that we check expression of the microRNA and transcription factor in different tumors, and especially in melanoma, we saw a very clear correlation. But in other types of cancer, we see also some kind of correlation between the two, indicating that this amplification mechanism may be a potential target for treatment. On the other hand, we discovered different microRNAs which are in fact inhibiting this time, not stimulating, not feed-forward activating, but inhibiting autophagy. These are all published here. You can check the articles if you would like to. So the basic idea is that, for example, upon starvation, these LC3 proteins, remember the LC3 proteins which are on the autophagosome membranes, they are lipidated and they are binding to the membranes. So when you label them or when you fuse them with GFP, you see these dots. So these dots, in fact, they labeled autophagosomes. So you see that here there are more dots upon starvation. If you count them, you see there are more dots. But then when we put the microRNA, the dots are decreasing. And we discover the targets of these microRNAs. And this is a complementation assay where we give back the target to prove that the target is relevant. You can see autophagy coming back here or here, okay? So we can do the opposite. We can inhibit the microRNA with some anti-microRNAs called antagomeres. You can see that here P62 is the autophagy receptor. You see its degradation upon autophagy activation because it's also going to the lysosome and degrading there itself. So when we inhibit the inhibitor, the microRNA, you can see that there's more degradation of P62, meaning that we stimulate autophagy by inhibiting these microRNAs. So you can play around with these molecules. And the bottom line is there's a gas and brake system where, in fact, autophagy signals activate autophagy. And if it's uncontrolled, this stress-activated autophagy, it can damage the cells because of different roles that, on vital organelles or energy and so on. So nature wants that you activate it and you put a brake on it. So the activation signal, here you see, for example, starvation, activates also the level of the brake, the microRNAs. And gas and brake system lead to increased autophagy, but not too much. So microRNAs are serving as a brake to control autophagy in the system. This is our model in relation to our discoveries. But these microRNAs, in fact, they also have a marker potential. So in the beginning, it was very difficult for us in Turkey to have access to clinical material because, hospitals and clinicians are more oriented towards clinical research. And there's, I believe there's still a gap with the exception of some centers, of course, there's still a gap between the clinical research and basic research in our country. And we hope to overcome this. And I'm working actively to help overcome it. But at that time, we didn't have access to patient material. And in collaboration with different clinicians from different cities, we could obtain a series of tumors where we checked, in fact, the relevance of our microRNAs as cancer markers. So we had access to colorectal cancer at that time. So we checked in a rather small series of colorectal cancer the level of our microRNAs. And then we discovered that, in fact, the microRNAs, stress-activated microRNAs, they are highly upregulated. And we compare these to, for example, carcinogenic antigen, CEA, which is a classical marker, or CA19-9. And we saw that the microRNA detection serves as potently as these markers, or even more potently, as a colorectal cancer marker. So this opened the way to a collaboration with Murat Kaya Yapıcı, who is currently a professor in Sabancı University and is affiliated with SUNUM as well. So we are now currently developing electronic-based nano-biochips for better and fast and cheaper diagnosis of colorectal cancer using our microRNAs. And I tried to patent. It's a very painful process, but it's taking a very long time, especially, depending on the different conditions. So especially in our field, there still needs to be some improvement and achievements in the field of patenting molecular biology or medical research. But still, we gave a fight and we still struggle, but we are trying to patent these molecules so that they will be the propriety of our country, and so we can, develop these tools in a more original and innovative way, and this will give us power. Okay, that's my approach. So these studies are ongoing. We have several outputs from this research, but I will present it maybe in the coming meetings. For example, the microRNAs which are inhibiting autophagy that I have shown you earlier, we saw that, in fact, in combination with chemotherapeutic agents, they have a potential to serve as an adjuvant treatment. What does it mean? So if you put the chemotherapeutic agent here, for example, etoposide, and you add the microRNA, so you inhibit the autophagy, you see that this is survival of cells, this graphic. So there are less cells which are surviving, meaning that you can kill cancer cells more by inhibiting autophagy in combination with the chemotherapeutic agent here. And this is valid for different types of chemotherapeutics here shown, or shown in other, also several data are available. So we said, can we use these microRNAs as a treatment strategy to treat cancer in our hands? But then microRNAs, as I mentioned, they are small RNAs. So you need gene carriers for this purpose. So we again turn to nanotechnology, and this was the beginning of a very long-lasting friendship and collaboration with Havva Funda Yağcı Acar from Koç University. Now she's appointed the vice president, vice rector of the university, and which is a big gain for, I think, the university. So she's an excellent scientist. And over the years, we collaborated, we started to understand each other, and we collaborated over creating some interesting nanoparticles which serve as drug carriers or gene therapy carriers. So here you see an example. These are quantum dots, which are, by the way, subject to the Nobel Prize of Chemistry, I think this year. So these quantum dots, they were covered with some polymers, and we could show that these polymers could easily deliver genes of interest inside cancer cells. You can see them shining here, and we also have evidence that the genes are entering and expressed. And we performed several studies. These are some of them only, using different nanoparticles. But it is my favorite is this particle, which is in fact based on iron oxide. So it's completely biocompatible because this iron oxide is used as a contrast agent for imaging purposes for many years now. It has also some other advantages. So you can follow the iron particles with magnetic resonance imaging, MRI. And you can also focus the beams trying to warm them, so to create a thermal therapy, like hyperthermia therapy. Increased temperature is also causing damage in the tissues. So we tested all this, and we see in our hands that MRI, you can follow these particles with MRI, and you can cause hyperthermia. This was in collaboration, again, with Hava Funda Yağcı Acar and Ali Koşar from Sabancı University and SUNUM. And we could observe these particles. Using these bases, we created some more complicated particles. We wanted to target these particles specifically to cancer cells. So we added an antibody. In this case, as a proof of concept, we added the HER2 antibody because we were planning to use breast cancer cells. And we labeled these antibodies with the red dye. So the particles, they look red, okay? On the other hand, this was another innovative step of these particles. We proposed and tested that AGO2 protein, which is a natural microRNA binding protein, could be used as a gene carrier. Gene carriers have these problems that they are all, most of them at least, are based on the different charges, because nucleic acids are negatively charged, and in general, they create cationic particles, so that the positive and negative charges will lead to accumulation of these gene fragments. But here we created a neutral particle using a protein which is binding to microRNAs. And on top of that, we saw in the literature that there were some indications, at least, that AGO2 proteins are responsible for freely circulating microRNAs in the bloodstream. So how can it be? Because microRNAs are RNAs, so RNAs are very fragile. So we also tested this in our laboratory, and we saw that, in fact, AGO increased the robustness of microRNAs and delayed significantly their degradation. We left them, I think, 3 weeks or so on the benchtop, and AGO significantly protected the microRNAs. So this was a good carrier, and this was the, one of the eureka moments. Okay. Then we added our microRNA. So, by the way, we went through the hard way. We patented the microRNA's medical use. At least we have the European patent now. And then this particle, we called it İzil Demir, because İzilemek, you can follow it. Demir is because of its iron oxide, and Mir at the end is also representing the microRNA, so it's a microRNA gene carrier. So I'm from Izmir also. This is another thing which is sounding, is İzildemir. So we call this İzildemir. Okay, so this is also currently patented, and the publication here is here. Okay, if you want to check it. This is also a very respectable high impact factor journal where we publish this work. So just I'll go over it briefly. So the particles are red. These red dots represent the particles. We injected these to nude mice, where we created a xenograft cancer model of human breast cancer. So we inject the cells under the skin of the mice. They don't reject because the human cells, because they have some defect in the immune system, these mice. They are very special mice. Because you cannot inject human cells to regular mice. They will reject it, okay? So we use immune deficient mice. This is an international model. So we created this tumor. You see it here, the tumor, okay? So now we inject through the tail vein the nanoparticles. The particles travel in the bloodstream, and they find the tumor. And this is a real picture taken by IVIS imaging system. It can show you the fluorescent image created by by the nanoparticles. So you can see exactly the tumor location very brightly, but the rest is not shining, okay? The eye is artifact, by the way. They have a red eye. Okay. Then we checked in the tumor whether, we check, of course, in the in vitro, in the cell lines and everything. You can see it in the paper, but a lot of work. But to cut the long story short, you can see here the control tumors where you see the nuclei staining blue. But then when we injected isildemir, you can see that the isildemir, the red nanoparticle, is accumulated inside the tumor very intensively and not in other tissues. We checked this also as well, of course. And in the tumor, we see that nanoparticle deliver this cargo, which is the microRNA. You see a significant increase in the level of microRNA when injected by isildemir in these tumors compared to control tumors. And we discovered them, as you remember, as microRNAs which inhibit autophagy. So this is an antibody which is recognizing lipidated, meaning autophagosome-associated LC3. So showing us the autophagosome intensity in these cells. You can see that this is very green. So, and when we put isildemir, the microRNA goes and inhibits its targets and inhibit autophagy. This is very faint, showing that, in fact, with this microRNA and the nanoparticle, we could downregulate autophagy in vivo, in living mice, in the tumor, and not in other tissues. We check all this. And these tumors are drug resistant, and these tumors become, now we expect them to become drug sensitive. Let's see that if it's so. So we combine isildemir with cisplatin. So what's the major problem with the chemotherapeutic agents? They go everywhere, okay? Look, we inject the green chemotherapeutic agent, they're everywhere. This is why, in fact, when a person is getting chemotherapy, they lose their hair, they have gastrointestinal problems. I mean, the worst cases, they have secondary leukemia, blood cancers, a lot of, a lot of things. So in the last, I think, maybe 30 years, people are trying to combine different drugs so to decrease the amount of individual drug that is used, but still the side effects are very severe, and it's a big trauma for the patient. So the best is to combine chemotherapeutic agents, I mean, with these innovative drugs, so that the drug will be more targeted and the effects will be more local, and they will be concentrated in the tumor and not in other tissues. So you can see that our drug is not accumulating in other tissues. It's only accumulating in the tumor. So this gives us specificity. And in combination with cisplatin, we observe that, in fact, the tumor size shrank significantly in a very short amount of time. And you can here see the graphics of it. This is isildemir together with the cisplatin compared to cisplatin alone at this time period. So we call this adjuvant treatment, okay? So in addition to chemotherapy, you add something next to chemotherapy and it's increasing the potential of chemotherapy. So this has a big potential. And targeted delivery is one component. Autophagy inhibition is another component. Ago usage is increasing stability. So it's an innovative drug, isildemir, okay? One more story is about tumor and microenvironment interaction. In the last, I would say, maybe more than 10 years, we are interested in this topic. And we are mainly interested in the interaction of cancer cells with fibroblasts, because you have in a tumor, in addition to mutated tumor cells, you have normal-like cells, including immune system cells, stem cells, and other types of cells, and also fibroblasts inside tumors or around the tumors, okay? And they are not neutral, they are not bystander, they are actively modifying tumor behavior, and in many cases, they are assisting the tumor cells to proliferate, to gain drug resistance, to metastasize, and so on. So we were interested in this interaction. The first study came from a collaboration with a very excellent group from South Korea, the group of Professor Cho, Yong-Kyun Cho. In this collaboration, we created together a system where we detected single cancer cell, single fibroblast interactions by using a patented original biochip that we created together. So this was a very long journey, of course, but it was successful, and now it's patented in the USA and other countries as well. So the idea is that this is the chip, in fact, how it looks. It has a membrane, and the sizes of dots are important. The diameter of these dots are important. And when you look at higher magnification, the cancer cell is red. You see that in the middle of the dot, the hole, there's the cancer cell, and this membrane, under this membrane, you have a carpet of fibroblasts, which are green. And these fibroblasts, when stimulated, they activate autophagy, and they have these green dots. So by this way, we measured autophagy and quantified and used TGF-beta as a tool. And by the way, at that time, it was not very well studied as an interaction partner and autophagy stimulator. This was also one of the important findings of this study, that TGF-beta activated, tumor-derived TGF-beta activated autophagy in fibroblasts, okay? So you can see that there's an increase, but then when we inhibit TGF-beta with antibodies or knock down TGF-beta in tumor cells, you can see a decrease in autophagy in these cells compared to here, okay? So, but then we didn't stop there. We said, okay, the interaction involves probably more than TGF-beta. Maybe there are some other factors. So we obtained, again, from Edgene, a very cheap set of cDNA, cytokine cDNA constructs at that time. This is a cDNA expression library, very basic library. At the end, in total, we have more close to 100 sorts of cytokines now. So we said, let's go, the shortcut. We pulled the cytokine genes, we transfected them to hex cells, and then we obtained the supernatant. So we're hoping that these hex cells will synthesize them and secrete them. And then we put them on fibroblasts, which have LC3 GFP, meaning that if there's autophagy activation, they form dots. Okay? So as you can see here, the dots are formed. And we discovered a factor, CTF1. The name is cardiotrophin 1. So it involves the earlier studies. Most of the studies involve heart biology, okay? Biology of the heart. So these are fibroblasts, but we said, okay, so what happens? Where is the context where cancer cell meets the fibroblast? This is the tumor microenvironment. So cancer cells are in contact with naive normal fibroblasts in the tissues, and they do something to them. They have some interaction and they change them, okay? And fibroblasts are important because they create the tumor environment. They have some interaction with cancer cells, but also they are the major cell type that secrete the extracellular matrix. So this matrix, the jelly, where the cancer cells or all the cells are residing, okay? They're important for wound healing and many things, as you know. So these cells, which are activated by cancer cells or are called carcinoma or cancer-associated fibroblasts or CAFs. So they support tumor growth, drug resistance, migration, invasion, and metastasis. And most of the studies, until recently, they concentrated on the interaction of cancer cells, but with fibroblasts from the other perspective, what the fibroblasts are doing to cancer cell. But how cancer cells stimulate fibroblast transformation or transdifferentiation to carcinoma-associated fibroblasts or CAFs was not studied well, and still there are some gaps. So again, to cut the long story short, we discovered that, in fact, CTF1 in a comparable manner to TGF-beta, stimulated cuff-like phenotype in naive fibroblasts. You can see accumulation of CTF1, actin, alpha muscle actin in response to CTF1 stimulation. You see that it's comparable to GGF-beta, and also transcriptional activation. Also, formation of stress fibers. You can see here that TGF-beta leads to activation of fibroblasts, and these stress fibers are formed, and alpha smooth muscle actin upregulation result in contractility of this fibroblast. And you can see here phalloidin staining, which is active cable staining, active stress fiber staining. And in a similar manner, you can see the same here with CTF1. So CTF1 is activating the fibroblasts. And we discovered also one more important thing, that autophagy is important for calf formation and fibroblast activation. This is also a very new contribution. And as I said, contractility, you can see that the collagen matrix, which is on the plate, it's contracted with TGF-beta, but also CTF1, shrinking the matrix because the cells are squeezing it. And autophagy is important. When you don't have autophagy, this doesn't happen. And also, there are some signaling issues. It's independent from IL-6. The effect of CTF1 is independent from IL-6. So what does it do, this interaction, and what's the role of cancer-secreted CTF1 in fibroblast communication? So we discovered that, in fact, this is, we confirm, in fact, the data in the literature showing that, in fact, cancer cells and fibroblasts, when they are together, migration and invasion. So this is migration of fibroblasts, this panel, and this is invasion. And this is migration, invasion of real cells from real patient-derived cells originating from patient tumors, fibroblast, carcinoma-associated fibroblasts. These are activated fibroblasts. In both cases, we saw that when in co-culture cancer cells with fibroblasts, you see activation of migration and invasion. So activation of metastatic, cancer spread capacity, metastatic capacity. And this is depending on CTF1 in our hands. So when we put the neutralizing antibody, you see that the metastatic capacity is decreasing in all cases. So CTF1, tumor-derived CTF1, is important for fibroblast activation and fibroblast-assisted cancer cell migration invasion, which can be a basis of cancer metastasis. So we further went on checking this in patient-derived tissues, tissue sections. You can see here that tumors which have high level of CTF1, they have high level of alpha smooth muscle actin and fibroblasts. So low level CTF1 expressing tumors, they have low level of fibroblast activation. And when we check these tumors, we could see that these breast cancer tissues, we could see that, in fact, a higher breast tumor size, but also, more importantly, lymph node metastasis. This is no lymph node, this is positive lymph node. Lymph node metastasis frequencies correlated with the expression of CTF1. In fact, underlining and confirming our in vitro results with cell lines. So CTF1 is important, tumor-derived CTF1 is important for activation of fibroblasts, activation of the tumor stroma, which is assisting cells in migration, invasion, metastasis to lymph nodes. So this is a prognostic marker as well in this sense. So briefly, I'm summarizing the data that I've shown you here. Fibroblasts, in fact, naive fibroblasts, when the cancer cells grow and they secrete factors like TGF-beta or CTF1, they are coming in contact with cancer cells and they are changing. So I cut the long story short, but you can check in the article. We discovered also the signaling mechanisms involved in CTF1 activated autophagy and fibroblast differentiation. So you can check this. We also perform mass spec analysis and so some metabolic changes and so on. So your autophagy is important. And these fibroblasts, they form these cables, they activate, they are their expression profiles are changing, and they become CAFs, cancer-derived, cancer-associated fibroblasts. So these cancer-associated fibroblasts are important for cell migration assistance and invasion assistance to tumor cells, as well as lymph node and metastasis, and possibly metastasis to other organs. Okay, so you can check all these articles that we published for the details of this study. Also, we have the patent application, I hope, which will be now final, it will be accepted soon, at least in the European Union. And all these markers, we check also their expression in the circulation in blood of the patients. So by this way, we try to discover novel diagnosis tests. And some of them also might be important for tumor growth or metastasis. So we develop peptides or antibodies, which can be used for, which can be tested for treatment purposes, but which also on the way can be tested to create original patented biochips using our antibodies, our peptides. And this work is ongoing in the frame of the 1004 Nanosis project, which is led by SUNUM, and I'm one of the leaders of this project. I think I'll skip this part because we are a bit late, I guess. But maybe in the next meetings, I will tell you about cancer dormancy. So just one word, maybe. You see the tumors here forming the cancer diagnosis. This is a PET scan. You see a lot of tumors. These black dots are tumors. This is a person, and this is the chest cavity. This is the lung tumors in the patient. Then after treatment, you see no cells. But then, in fact, you should see cells, but you cannot see them with the current detection methods. So you see all these red dots represent cells which are present in the body, but which are not detectable. So mice experiments and bone marrow analysis in breast cancer patients show that, in fact, a cancer patient who has been treated for cancer, after treatment, they still have around at least 3,000 to 6,000, maybe thousands of cancer cells which are residing in their tissues. They are not dead cells. They are like potential time bombs that can anytime reactivate. So unless we get rid of them, we cannot get rid of relapse or recurrence. And when they come back, these cells, when they grow again, and we don't know how they switch off and how they switch on, they are very dangerous. They can cause, again, tumors, but this time more aggressive tumors. And in general, these are the metastatic or non-metastatic. These tumors which come back are killing the patients. So we performed very large screens, discovered many genes, and we have couple of very, very interesting studies which are ongoing. And I hope to be able to present them to you in the next meetings. And we also think about, how to target these cells. So you can follow them up. And then when they activate, before they activate, you can kill them. But for this, you need detection methods. And we are also working on that because the current detection methods cannot detect tumors smaller than half a centimeter or 2 millimeters. Half a centimeter or 2 millimeters is already a quite big tumor. It's like a rice, size of a rice or something, or half of a bean, for example, or quarter of a bean. So it's not a small tumor. And smaller than that, we cannot, in many centers, we cannot detect. And in the best centers, you can detect maybe 2 millimeters to half a centimeter. So single cells are there and they are staying. If you could see them, this would help us to follow them. Or one risky approach is to activate them and to make them visible and then kill them. But then this is, of course, ethically, I don't know if it's possible. But the best is to find drugs which eliminate these non-dividing cells. Major problem is currently used cancer drugs are mainly killing cells which are dividing faster, because you don't want to kill the normal cells of the body. You target cells which are dividing faster. So most drugs cause DNA damage, microtubule toxicity, or reactive oxygen, which will lead to those. So chemotherapy and radiotherapy is causing all these changes. But these cells which are sleeping, because they are not dividing, they are resistant to these treatments. And in our hands also, we saw that they are completely resistant to chemotherapeutic agents, cancer drugs. So we are also searching for ways to kill these cells in our systems. Okay, just a few words maybe about Nano-Bio4Can. This is a European Union Marie Sklodowska-Curie action. I'm also part of it, and this is led by SUNUM. These are the websites. They are in preparation now. You can check them, and there's this information help desk here. The project is very important, and one of the main reasons why I'm contributing to this project is to bring forces together. This is another aspect in which we are unfortunately weak in Turkey. So there are individual success stories, but together, working together and creating together and achieving together is still a weak side of the research in Turkey. And lately with 1,004 projects, we are trying to achieve this goal. But the code of the project is 1,004. This brings together, tens of universities, centers, companies together around the project. And there are several big projects and some small projects and so on. But this COFUND project, I think is very important because it brings together SUNUM, the Nanotechnology Center, İBG, İzmir Biomedicine and Genome Center, TÜBİTAK Marmara Research Center, which is one of the major governmental research centers, research and application centers in Turkey, and also Koç University and Kuttam Translational Research Center, which is inside Koç University, both in the hospital and in the Rumeli Feneri campus that you see here. So all these four groups are participants to this project, and they are supporting different postdoctoral scientists who will join this project, okay? The main focus is to use nanobiotechnology for innovative therapeutic treatment approaches for cancer. And several aspects of nanotechnologies for cancer are focused here, from drug development, drug delivery systems, targeting system development, testing and validation systems, etc. We had a very thorough evaluation, and we prepared the project meticulously so that we will answer these requests. And of course, also for our high standard, we had to do this. So we have a very strict and very well planned, very well framed EU standard evaluation system. The most important thing maybe I would say here is that the candidates, these postdoctoral candidates who want to work in this project, before they apply, they have to be sure that they should not have resided or carried out their main activity, work or studies in Turkey for more than 12 months in the last 3 years, immediately before the call deadline. So students who are currently working and studying in Turkey, they are not eligible unless they spend this time of period abroad. So the idea is also to draw international scientists to contribute to this project, okay, and to give equal opportunities to everyone from Europe and outside Europe. And this is not a regular postdoctoral fellowship. This is something way beyond that, because we have planned several hands-on schools, industry days, open seminars. There will be intensive training, different courses you see here, schools on particles, medicine, drug. Always in the frame of nanobiomedicine, and also in universities in Turkey, in centers in Turkey, drug companies in Turkey and abroad, universities abroad, centers abroad. This is a very large consortium. But the main benefiters are these four institutions, okay? But there are several different companies, institutions, also universities and institutions abroad, in Europe and in the United States, they are heavily contributing to this project. These are the deadlines, tentative deadlines, so they might change. But if you follow through the website, you will have more information. Okay. So with that, I'd like to thank you for your patience and interest to our research. And I would like to thank also our sponsors that you see here over the years. Our collaborators, some of our collaborators are active collaborators. Some of our in the past years we continued collaboration, and these are the current members of Gözüaçık Lab. We are celebrating our 18th anniversary now. The lab, I hope it's reach maturity at the 18 years. I believe that it reached maturity because 18 years is like also adulthood. This is the website, gözüaçıklab.com. Okay, this is our main website. There are several websites. You can check our research, our activities, and different publications over here as well. With that, I'd like to thank all these supporters and collaborators and students, and I would like to thank you for your interest and patience. Thank you.
Hocam, thank you for your presentation. We already passed our time, but we can take a couple of questions. You can ask your question directly, Devrim Hoca, or write them in the chat box.
You're also most welcome to contact us by email, and if you have any questions, I'd like to, I'll try to answer them also through email if there are no questions here. You can also contact us. You can also, there's the postdoc project. You can come work with us as a postdoc, or you can come work with us as a PhD student. Follow the deadlines of all these institutions if you want to work with us and our collaborators, of course.
Hocam çok teşekkür ederiz. Vakit ayırdınız. Verimli bir webinar oldu.
Teşekkür ederim. Biraz uzun sürdü ama bence değdi. Çünkü
Güzel
çok
oldu.
Nanosis'te ben
Nano-Biyoteknolojilerden de
Nano-Bioforken'i
bahsettiniz. Çok
anlatmak
iyi oldu.
istemiştim. Umarım faydalı olmuştur arkadaşlara da. Yani son cümle olarak şunu söyleyebilirim. Gerçekten Türkiye'de araştırma yapmak zor ve zahmetli. Ama yani ben şimdi uzun yıllarda yurt dışında kaldığım için birçok arkadaş da aynı şekilde. Ama buradaki kazancımız, buradaki öğrencilerin enerjisi, buradaki öğrencilerle yakaladığımız sinerji, buraya katkılarımız bence çok önemli. Yani hep birlikte burada güzel şeyler yapacağımızı düşünüyorum. Hem Türk hem yurt dışından bize gelen misafir öğrencilerimizle beraber ve araştırmacılarımızla güzel bir ortam oluşturmaya çalışıyoruz. Bu bahsettiğim 4 tane kurum, SUNUM, İBG, TÜBİTAK MAM ve KUTTAM. Bunlar gerçekten çok iyi imkanlara sahip. Yani yurt dışındaki en iyi yerlerle yarışabilecek imkanlara sahibiz. TÜBİTAK gerçekten çok destekliyor. TÜSEP de var ayrıca ama TÜBİTAK gerçekten çok destekliyor. Ve yani bu genel işte ekonomik koşullar vesaire, pandemi, şu bu onlarla alakalı zorluklara rağmen çok büyük bir çaba var. Hem öğrencilerimizi hem araştırmaları desteklemek için. Hep birlikte mücadele etmek durumundayız ve Türkiye gibi bir ülkenin, böyle büyük ve güzel bir ülkenin bence bilimde de çok daha güzel yerlere gelme potansiyeli var. Onu da umarım buradaki arkadaşlar ve diğer arkadaşlarla hep birlikte başaracağız. Vazgeçmek yok diyorum. Hep beraber çalışmaya devam edelim, uğraşmaya devam edelim. Biz her zaman daha senior olarak her zaman daha buradayız, arkanızdayız. Desteklemeye, yardımcı olmaya çalışıyoruz. Elimizdeki imkanlar dahilinde, zamanımız dahilinde. Çok tekrar teşekkür ediyorum. Sağ olun.
Biz teşekkür ederiz. Chat'ten de katılımcılarımız teşekkürlerini ilettiler.
Sağ olsunlar.
Katılımcılarımıza da çok teşekkür ediyorum. Haftaya yeni bir konuyla tekrar karşınızda olmak üzere. Hoşça kalın.
Çok sağ olun. Teşekkür ederiz. Hoşça kalın Nokta Hanım. Hoşça kalın herkese.